Hi there, this is Professor Schimmold from Crafton Hills College. Today I want to talk to you about classification, and I'll make some general remarks about classification of all of the organisms that live on planet Earth, and then we'll talk specifically about how is it that we make classification decisions for prokaryotic organisms. Okay, so you've got a chapter outline, and it's got some opening points. Let's go ahead and review those together. So classification, that refers to taking organisms, a group of organisms, and dividing them into smaller groups. These smaller groupings are going to reflect relationships between the organisms. Now, classification is also referred to as taxonomy. The root word taxa means category. All right, now the most basic division when we're talking about classification is between prokaryotic and eukaryotic organisms. And we're going to take a look at a phylogenetic tree of life, and we'll also look at a nice outline of the system of classification that we're going to use this semester, which is called the 3-domain 4-kingdom system. All right, back to that list on the first page of your outline. Organisms are placed into groups with other organisms having similar characteristics. For example, all of the plants would be in a group called Kingdom Plantae, all of the animals in a group called Kingdom Animalia, etc. The word phylogenetic, that refers to the evolutionary history of an organism. So who's related to whom and how closely so, who evolved from whom. And phylogenetic history refers to the evolutionary history of an organism or a group of organisms. Now, taxonomy, classification, also useful for identifying known organisms. Let me give you a couple of examples. Let's say you have an eye infection, and we're going to go ahead and we're going to take a sample and we're going to cultivate it on a microbiological media in the laboratory, and once the organism, the bacterium, I'm assuming for this conversation, has grown up, then we will perform a number of tests on it. For example, we will determine is it gram-positive or gram-negative, cocci, bacilli, etc., etc. We can gather this information and then apply that to, or compare it, I should say, to the classification of known organisms, and that information can help us determine the identity of the organism that was causing your eye infection. Okay, now on the bottom of the first page of your outline, you have something that looks like this, and this is referred to as a phylogenetic tree. And this is what taxonomists have put together to try to show the evolutionary history of all organisms that are residing on planet Earth. Now, you may or may not have noticed at this point that this does not include the viruses, because in many ways we just don't know what the heck to do with them as far as classification goes. Now, so if you take a look at that phylogenetic tree of life, you'll see that it's broken into 3 main groups. The domain, and those are domains. First is the domain known as bacteria, also known as eubacteria. EU. I may have mentioned this before. If you put EU in front of a word, it means true. And then another domain that includes only bacteria, and that would be domain Archaea. And then all of the eukaryotic organisms are in their own domain. Now, I don't expect you to memorize this phylogenetic tree of life, but I do want you to understand how it works. So I've made a very simplified little sketch, not entirely accurate, you guys, just to try to make a point. And so what we're looking at here is down here, this would be at the bottom of the tree, that would be the first form of life on planet Earth, which we assume was a very simple, even for a bacterium, a very simple bacterium. And then each of the branch points represents a major evolutionary event. It could be like the ability to use oxygen in your metabolic pathways. That's just an example. But anyway, so each point represents a major evolutionary event, and you can see how beginning with our first prokaryotic cell, that lots of evolutionary events have occurred, obviously, and they have led to the development of these three large groups and then the smaller groups. Now, when we look at the branching points, their relationship to each other is going to indicate to us when that evolutionary event took place, and if we are comparing where a group of organisms are in relation to another on this tree, the closer together the branches, the more closely related they are. That probably didn't make a lot of sense, so let me try. Now, let's say you can see that I've applied some numbers to many of these branches for this discussion. So let's say if we compared organisms that are in these two groups, the ones labeled 4 and 5, the branch and the branching point and the branches are very close together, so those organisms are very closely related, as opposed to comparing 4 to 15 over here. You can see that a lot has happened, and those organisms will not be as closely related. Okay, let's go ahead and move on. Enough of phylogenetic trees, at least for the moment. All right, so let's go ahead and talk for a moment about nomenclature. And nomenclature refers to assigning scientific names to organisms, not just bacteria, all organisms. Once they've been identified and so forth, they are given a scientific name. Now, we use a system that was developed by Carolus Linnaeus in 1735, and we refer to it as being a binomial nomenclature system, meaning two names. Now, the two parts of a scientific name are the genus name and the species name, and there are some rules for writing scientific names. Here are the rules. First of all, the genus name is always capitalized. The species name is not capitalized. And then if you are writing by hand like this, if you're writing by hand, you would individually, like this, individually underline the genus and the species name, genus name, species name. If you are word processing, then you can choose. You can either underline the names or you can italicize them. You don't do both. You do one or the other, underline or italicize. All right, so genus name capitalized, species name not capitalized, and both names are individually underlined. Now, what's in a name? In some cases, there is some potentially useful information in an organism's name, and I'm going to talk about a bacterium for this conversation. All right, so back to Staphylococcus aureus. So Staphylo means that the cells are arranged in clusters. Sometimes they're referred to as grape-like clusters. And I'm talking about if we were to view this bacterium microscopically, put it on a microscope slide, air dry, heat fix, stain it, let's say gram stain or whatever, you would see that the cells are arranged in kind of grape-like clusters. And coccus or cocci, coccus is singular, cocci plural, those are round bacterial cells. So if you take a look at this little chicken scratch drawing here, this is supposed to be your field of vision through a microscope, and you've got round bacterial cells arranged in grape-like clusters. All right, now a little bit about information in names. So we already know that Staphylococcus aureus, that the cells are round and that they're arranged in grape-like clusters. Let's go ahead and talk about the species name, and then I'll kind of circle back for that other part. Aureus, derived from, I think, Greek rather than Latin in this case, meaning golden. So this organism is sometimes referred to as the golden staph. Here's what I mean. Now, if you were to cultivate, if you were to grow Staph aureus on a plate containing tryptic soy agar, the colonies that would grow would be pretty much pure white. If you were to inoculate that very same organism, Staph aureus, onto a different type of media called blood agar, and that's just TSA with some sheep blood added to it, sometimes, not always, but sometimes the colonies that grow will be a very pale yellow color. And so an experienced microbiologist could see those little pale yellow colonies on a blood agar plate and have a pretty good idea of who they were dealing with. Now, Staph and cocci doesn't sound like a lot of information, but a physician or a microbiologist, just by knowing the genus name of the organism, they would have a pretty good idea of what antibiotics would be appropriate and effective to treat an infection with this organism. So there really is some important information in a name. Now, other times, scientific names may be honoring or reflecting the individual that discovered an organism. For example, you've heard of E. coli, the bacterium E. coli. That's short for slang for Escherichia coli, named after Theodore Escherich. I'm not positive if I'm pronouncing his name right, but I think it is. And so he was given the kind of dubious honor of having an inhabitant of the lower GI tract named after him. And in other times, scientific names really don't contain much useful information at all. For example, there's a species of the bacterium Salmonella named Salmonella jordani, which I remember reading about. The microbiologist that discovered that bacterium was a Michael Jordan fan. So there can be information, useful information in scientific names and other times not. A little bit more about Staphylococcus aureus. And I bring this up again because this is such an important potentially serious pathogenic organism. All right, what did I write on here? Oh, sure. There are strains of Staph aureus that have been named, they call it MRSA, methicillin-resistant Staphylococcus aureus. And this is a highly drug-resistant strain of the organism, resistant to all known antibiotics except one called vancomycin, developing some resistance problems there. There are some new antibiotics coming down the pipeline, so hopefully some of those will help us with this organism. Also causes a very common form of food poisoning, staphylococcal food poisoning, sometimes erroneously referred to as the 24-hour flu. There's no such thing. If you have had something to eat and 1 to 6 hours later you find yourself, in the bathroom, it's not a coincidence. You've been food poisoned. Staph aureus produces a category of toxin called enterotoxins. They're very heat stable, and it's the enterotoxin that's going to cause the symptoms of that type of food poisoning. Let's see, Staph aureus, I'm trying to read backwards through this, also causes a disease called toxic shock syndrome. We'll talk about it later this semester, and skin infections known as boils, which are quite painful and unpleasant. So those are just some of the things that Staphylococcus aureus can do for us, or to us, I suppose. Okay, where are we? All right, now, as you might imagine, or maybe not, there are so many thousands and thousands of species of bacteria and other forms of life that pretty much at a constant pace, new organisms are being identified and named. And when we are referring to bacteria, any new additions like newly identified organisms, or perhaps name changes, because as our technology is improving, we're having to kind of reshuffle, reclassify things. I'll address that in a little more detail in a few minutes. But anyways, there is a very exciting publication called the Journal of Systematic Bacteriology, and new additions and changes are published in that journal. Okay, it's not a very interesting journal, but an important one. Okay, now let's go ahead and talk about taxonomic systems or classification systems, because there are a number of different ones, and pretty much anybody that does taxonomy has their own opinion on, if you took all of the life on Earth and you attempted to divide it into groups and create these categories, everybody has a different opinion about where the division should be. So what I've done is I have picked a system, and that's the one that we're going to use this semester, and you should have this in your notes. It says 3-domain, 4-kingdom system of classification. I want to go ahead and review this with you. All right, I've already named for you the 3 domains. First is domain Archaea, And the word Archaea means the ancient ones, because of the two main groups of bacteria, these are the more primitive variety. They are prokaryotic, the Archaea are prokaryotic, and they do not have any peptidoglycan in their cell walls. Remember we talked about gram-positive, gram-negative bacteria. Well, these guys, the Archaea, they do not have any peptidoglycan, and taxonomists decided that having peptidoglycan or not having peptidoglycan was important enough to divide the bacteria into those two large domains. All right, now the Archaea often live in extreme environments. Let me give you some examples. Three examples of Archaea. One would be the halophiles, and halo means salt, phile means lover. These bacteria live in highly saline environments. Not as the ocean is not salty enough. I mean places like the Great Salt Lake, Salton Sea here in Southern California. Sometimes out in the desert you see salt evaporators where extreme temperatures are pulling water and salt up to the surface of the ground where it can be scraped off and harvested. Sometimes you'll see halophiles growing in those environments. So that's a pretty extreme environment, I think. The Methanogens, these bacteria typically live in swampy areas. They help to decompose organic materials like dead plants, dead animals, etc. And they release methane gas as a metabolic byproduct. And then the thermophiles, heat lovers. These bacteria live in extremely hot environments, often having a very high sulfur content. I'm talking about surrounding volcanic vents deep in the ocean, right? So there's another example of a very extreme environment. Okay, second domain, bacteria or eubacteria. And these are what we call the true bacteria. And we're referring to true bacteria as those cells that contain a little bit or a lot of that peptidoglycan. Most eubacteria have cell walls. There are some exceptions to that. We talked about that earlier in the course. Let's see, what else do I want to tell you about them? I guess we're good on domain eubacteria for now because we're going to be spending quite a bit of time looking more closely at them later. And then domain Eukarya. EU, true, karya means karyon or nucleus. These are the rest of the organisms that all have cells that contain a nucleus and organelles. All right, so we're going to take domain Eukarya and we're going to go ahead and divide it into 4 smaller groups, and we're going to call those groups kingdoms. And those kingdoms are, and let me tell you just a little bit about each one. First, I wanted to mention kingdom Protista, also known as the Protozoans. I mean critters like amoeba and euglena and paramecium. They are unicellular. Most are heterotrophic, meaning they're going to bring nutrients, pre-made nutrients in to their cells for metabolism. Others are autotrophic, meaning they have chloroplasts and they can use photosynthesis to produce sugar, their own nutrients. Kingdom fungi, now this is a very diverse group. They're all eukaryotic, all made of eukaryotic cells, of course, and some fungi are unicellular. We call them the yeast. Others are multicellular. That would include things like molds and mushrooms and mildews, all right? And molds are fuzzy, filamentous, right? Mushrooms, what we eat and what we see is the part of the organism that grows above the surface of the substrate. It's called the fruiting body. And mildew tends to be kind of a powdery substance. Now the fungi are not capable of photosynthesis. They are not plants. And what they do is they absorb their nutrients through their outer coating, which is often called a cuticle. All right, kingdom plantae, the plants, multicellular, use photosynthesis to produce their own nutrients. So we say they are autotrophic. And then kingdom animalia, that's the kingdom that we are classified in, multicellular, heterotrophic, and the animals ingest their nutrients, which means take them in through a specialized opening. Usually that's called a mouth. although sometimes it's simpler than that. All right, let's go ahead and move on. I want to just kind of go over with you an example of classification, and we're going to go ahead and review the classification of the domestic dog, just because it's something that we're all familiar with. And first of all, let's take a look at what we call the hierarchy of classification. Now, that means the ordering of groups, of taxonomic groups, from largest to smallest. And I like to use this analogy. Why don't you think of this hierarchy of classification as an inverted triangle with domain occupying the top and species occupying the bottom, and all the other categories are in between here, right? So something like this, okay? So domain is the largest of the taxonomic groups, and it is the least specific group. Here's what I mean. If the only classification information that we know about an organism is the domain it's a member of, we don't know a whole lot about that organism. All right, and so then it would go, let's say we're talking about the dog, domain, kingdom, so forth, okay, down to species. If we knew all of the taxonomy information about the dog, all the way from domain down to species, we would know quite a little bit about the characteristics of the dog. Now, there are taxonomic categories that are actually below species, for example, subspecies, biovars, serovars, but we don't really need to include those in this conversation right now. So let me talk to you about what I expect you to know and what you don't necessarily need to know. First, you need to know the hierarchy of classification in the correct order. So domain, kingdom, phylum, class, order, family, genus, and species. Now, when I was a first-year biology student in 1975, maybe, they had not yet developed domain, so it was just kingdom through species. And they taught us this silly little thing to remember it, and, well, I still do, and that is kings play chess on fiberglass stools. Okay, whatever, it worked, right? I still remember it. Well, domains have been added since then, and one of my biology students some years ago taught me a way to memorize that. And it goes, dumb, kinky people come over for group sex. No judgments here, people, just trying to find ways to remember things. Okay, now a couple other notes. The group known as a phylum, which is right under kingdom, the plural form of that word is phyla, and the group known as genus, the plural form of that word is genera. Okay, so taking a look at the dog. Now, okay, so I expect you to know that hierarchy of classification, and I don't expect you to memorize the classification of the dog, however, and you'll have to remind me of this, if you are able to correctly write out the classification of the dog, including the hierarchy of taxonomy, on the back page of your next lecture exam, I will consider tossing some extra credit points in your direction. So you'll just need to remind me of that. Okay, so classification of the dog. First of all, member of domain Eukarya. Okay, and we know what that means. And we also know what members of kingdom Animalia have in common, right? They are multicellular, eukaryotic cells, ingest their nutrients. All right, so the kingdom can be broken down into several phyla. And the phylum in which the dog is classified is called Chordata, and that refers to animals with a notochord. Oh, look it up if you're interested. But typically, not always, but typically these animals will also have a backbone. All right, the class that the dog is a member of is called Mammalia, and those are chordates with hair and mammary glands. So you can see as we're moving down our inverted triangle, we're becoming more and more specific about the characteristics of the organism. Order is Carnivora. Those are mammals with similar teeth, and I'm referring to the shape of their teeth. Carnivores have teeth that are designed to rip and tear flesh, not really very efficient for prolonged chewing. The family is Canidae, and those are carnivores with 42 teeth. The genus is Canis. That includes things like domestic dogs, wolves, coyotes, jackals, and dingoes. and the species is Familiaris, the familiar or the domestic dog. Now, you never write a species name so that it stands alone. And another rule for writing scientific names is, let's say you're writing a research paper and you're talking about Canis familiaris. The first time that you mention the organism in your writing, you would want to completely spell out the genus and the species name. And then subsequent times that you mention that same name, you can abbreviate the genus name, but you must always write out the species name. Okay, I've got a note here in your outline. Now, I talked about the Journal of Systematic Bacteriology before. That is a journal that's published, I don't know if it's monthly or quarterly. But anyways, there is a book called Berge's Manual of Determinative Bacteriology, 9th edition. It's not a new book, but it's still widely used. And this book can be used to find way more information than you would ever really care to know about both gram-positive and gram-negative bacteria. Nothing about any other type of organism, nothing about fungi or protozoans. This book only includes information about bacteria. Okay, I think I'm going to go ahead and break here, so look for a part 2 coming up shortly. Thanks for listening.